So we're going to start the first session. And the first presentation is by Yingping Wang. Come on up, Yingping. Genetic locus associated with bleomycin induced drug fibrosis in mice. Take it away. And if you want to give a micro introduction to yourself, that's fine too. Thanks.
Good morning, everyone. I'm Yingping, and I'm a second-year graduate student from University of Colorado. Thanks for giving me this opportunity to discuss my project. So today I'm here to talking about genetic locus associated with bleomycin-induced lung fibrosis in mice. This study focuses on idiopathic pulmonary fibrosis, IPF, and it's a condition that some people may not be familiar with or fully understand its severity. So imagine waking up every day and struggling to breathe and knowing that you have an incurable disease. This is a harsh reality for millions of people worldwide who suffer from IPF. It's like a lung puzzle that things just don't fit right, like the bronchi stretch and the alveolar remodel endlessly, and the scars make the lung difficult to do its work. And what even more challenging is that there is limited treatment options. So individuals typically only live 3 to 5 years with this condition. IPF has several recognized risk factors, with the most significant being age, especially in males aged 50 years and older. And cigarette smoking is another prominent risk factor. Exposure to some environmental pollutant and occupational hazards, such as the mental dust, can also increase the likelihood of developing IPF. Genetic factors, including specific gene mutations, like those in the MAC5B gene, also play a significant role in the development of this complex and incurable disease. Understanding these genetic factors is crucial for early detection and potential treatment options. We have sequenced across the original 10 GWAS loci in IPF cases and non-disease controls, and I have identified these 10 common variants that in aggregate account for at least 40% of the risk of IPF. And among all these common variants, MAC5B promoter variant, this one, was the strongest genetic risk factor for the development of IPF. And it's a gain-of-function variant. It will cause the overactivity of MAC5B gene and cause the overproduction of the mucus in the lungs. And it's the key feature of the IPF. So individuals with this variant will have the increased risk of developing of IPF. However, not everyone with this variant will develop this disease. While the minor allele of MAC5B is present in around 20% of non-Hispanic whites, but the IPF occurs in far less than 1% of the population, and there is no clear explanation for the low penetrance of this allele. Like I mentioned above, IPF is a complex and multifactorial disease. So individuals who carry this MAC5B variant may also need to have other genetic variations or be exposed to some environmental triggers to develop IPF. And also, the MAC5B experiment is exact mechanism by which it contributes to IPF are not fully understood. Even though we know that it will lead to the overexpression of MAC5B and the overproduction of mucus in the lungs, but how it interacts with other genetic factors is complex and not fully elucidated. So if we understand the genes and that control MAC5B and its role in lung fibrosis, we can learn more about why IPF happens. The 8 founder mouse genes of the diversity outbred mice all carry the risk T allele at this orthologous locus, but we found they show varying association between the MAC5B expression and lung fibrosis. Here we use the bleomycin to induce lung fibrosis in mice, and after 3-week intratracheal bleomycin administration, The first column is the expression of MUC5B, and the later 3 columns are some phenotypes reflecting the lung fibrosis. So we can see that there are some strains show low MUC5B expression and minimal lung fibrosis, but some others are high MUC5B expression and extensive lung fibrosis. And there are also some they didn't show any clear relationship between them. So this study suggests that other factors may work in concert with or potentially independent of MUC5B to influence bleomycin-induced lung fibrosis. And these strains and their recombinant inbred crosses may prove helpful in identifying the genes and transcripts that interact with MUC5B and influence lung fibrosis. Diversity outbred mice are created from these 8 founder mouse strains, and due to the diversity genetic composition and the diversity of these strains in bleomycin response, they offer an excellent resource to identify genes and transcripts that involved in MUC5B-associated lung fibrosis. And high-resolution CT scans are used to diagnose IPF, and now there are ways to quantify features of lung fibrosis in mice and humans from the CT images. So based on all the information presented above, currently we are conducting GWAS and EQTL analysis in mice with bleomycin-induced lung injury. And in addition, we have already gathered human GWAS data. So after we got the results from mice, we'll assess the relevance of genetic and transcriptional changes observed in mice to humans with IPF, aiming to deepen our understanding of this condition and explore whether there are other genetic factors regulate the mechanism of MUC5B and influence the development of IPF. So the hypothesis is genetic variants other than the MUC5B influence the development of lung fibrosis. To test this hypothesis, our study comprises 3 parts. So first, we conducted QTL mapping for MUC5B protein expression and lung fibrosis, and subsequently the RNA-seq data will be added to this study to analyze the differentially expressed transcripts and EQTL. And finally, we'll determine the generalizability of the genetic and transcriptional changes we observed in mice to humans with IPF. We computed 91 quantitative CT indexes to assess the bleomycin-induced lung fibrosis in 546 DO mice. All these mice underwent the intratracheal bleomycin administration at the 12th week to induce the lung fibrosis, and they were harvested and phenotyped 10 weeks later. To assess the lung fibrosis, we used micro CT scans and used a technique for automated lung segmentation from these CT scans, and then use a pyradiomics software library to compute various radiomics features. Finally, we use this data index to reflect the extent of lung fibrosis. And totally, we have 91 CT indexes computed like this, and use this data to do the QTL mapping. For the genotype, all these mice were genotyped using the GigaMoga array and got 143,000 informative markers for QTL mapping, and the genetic loci was identified using our package QTL2. And through this analysis, we found a QTL peak on chromosome 7. It located like 9 megabits from the MUC5B gene, and it influencing totally 29 CT indexes and shows significantly association with 4 of them. So our boss was very interested in the entropy related indexes because the lung fibrosis are characterized by heterogeneity due to the scarring. Like if we have a scar on some tissue, it will show the intensity variability among all this tissue. So the word entropy appears to be particularly relevant to this kind of heterogeneity. And indeed, it is an index used to quantify the CT images and measure intensity variability within the image neighborhood. So here I took joint entropy as an example to show this QTL support interval. In this region, these red points are some SNPs with the highest load score, and all these top SNPs comes from stream PWK. It contains 10 genes. From our previous human data, 6 of these 10 genes showed a significant differential expressed in IPF cases versus non-disease controls. The stream PWK shows the strongest QTL effect within this genomic region, with a notable positive impact on joint entropy and negative impact on joint energy. And previous study has linked entropy negatively and energy positively with radiation-induced lung injury. So combining all these results, our conclusion is the allelic effect from stream PWK on this locus may reduce the bleomycin-induced lung fibrosis, and maybe it interacts with MUC5B or act independently. And this conclusion is aligned with our previous study identifying PWK as have the low MUC5B expression and minimal lung fibrosis. And currently we are conducting additional mapping to identify fibrosis protective genes, and the RNA-seq and eQTL analysis is upcoming. After we got all these results, we will assess the applicability of genetics and transcriptional findings in mice to humans with IPF, and finally validate some candidate gene function. So the above is the current stage findings of our study. Thanks for listening, and I hope this report have provided you with some valuable insight and information on our study. Thanks. Happy to take questions.